Hello and good evening, everybody. Please confirm if you can hear me. Good evening to one and all. Welcome to the free workshop on GATE 2020 strategy. I hope my audio is clear. Kindly confirm in chat if you can hear me. Welcome to the workshop, Nilomani, Bablu, Mansi, Prita, and all of you. Good evening, and I hope all of you are able to hear me clearly. Nivedita, can you hear me? Okay. All right. So we will be starting our workshop shortly. Also, kindly confirm if you can see me too. I hope I am visible as well. We will be starting the workshop sharp at 7, and our experts are right here live to answer all your queries in chat as well as throughout the webinar. So feel free to interact among yourself as well as with the teachers live, and they'll help you. Okay. So before we started, we have put up a small poll over there where we have mentioned, have you given GATE 2019? Please answer there. I can see that a lot of you are going to write GATE 2020 for the first time, and majority of you have written GATE Life Science. Very less people have written GATE Biotech, and we are really excited to answer all your queries today. So before I get started with the workshop, I would like to give you a brief introduction about the GATE exam. Of course, you all know GATE exam. GATE is conducted every year in the month of February. The notification comes somewhere around October, right? And you will be given a chance to apply. Do you have some age limits? Of course. Now, the merit or demerit of this exam is it is conducted only once in the year. So you have to really wait for 1 year to re-attempt it, okay? And that is why you must strategize perfectly, plan perfectly, and then execute, okay? So we're going to talk about the planning, execution, everything, okay? So now I will start sharing my screen. Please confirm if you're able to see my screen. In the meanwhile, I will also end the poll so that all of you can see the results. So 11% people have written GATE Biotech, 20% who are attending today have written GATE Life Science, and 68% of you have, not written GATE at all. So of course, we are here to help you. So today's workshop is all about GATE 2020, a fantastic year which is about to come, and we must prepare for it. So let us start today's workshop with a quotation by George Lorimer, which says, "You've got to get up every morning with determination if you're going to go to bed with satisfaction." And this is very, very apt for GATE as well as CSIR NET exam. You must get up every morning with the determination of qualifying this exam. So most of us, when we start studying, we go straight. Now, what is going straight? That is, you will first think, okay, let me study so that I can qualify GATE exam. That is the wrong approach. The right approach is, I have to get highest rank in GATE. Now, what all things I need to get there? Okay, so this is how you have to think. Also, you should not give a damn on what people say. Because people will come and say, oh, you've done MSc or BTech and still you have not got placements, so they will laugh at you. Then they will challenge you. If you are really talented, go get a job, high paying job. Then they will appreciate you when you're working hard for these exams. And finally, when you qualify with a high rank and percentile, then they will set you as an example. Okay? And this is what happens with most of the students. So don't give a damn to others. Remember, you are a seed, right? Now, what is a seed? Seed has got the potential. You remember, we studied Turgor pressure. So the seed has got a potential to do great things. You are that seed. So you have got tremendous potential. And you can see in this image right here what is happening. So the seed grew into a tree. Correct, guys? Can you see this? Crack has come and it grew into a tree. That is what a seed's potential is. So you must increase your potential to qualify GATE exam. You must challenge yourself. See, you have mastered all the subjects of your BTech or MSc, but that is not going to increase your potential. You must challenge yourself with tougher questions, more tough questions. You must interact with more experts and then solidify. You must begin your day with a get to attitude and a have to thought process. You must get to work or get to study. Okay, whatever stress comes, we always think that okay, preparing for GATE and CSIR net is stress. Okay, but it is not stress. It is in fact blessings. If you prepare properly, this opportunity will come to you. You will qualify with a top rank. And like I said in the beginning, don't think that I have to study to qualify GATE. Think I must get the highest rank in GATE. Now, what all things I need to qualify GATE? This is how you have to think. You must change your thinking process. So what we have to do is to plan, and then obviously you have to execute. And if you are joining Biotechnica's coaching, then we'll help you execute. And finally, we will win together. Like you can see, this is the practice phase. In the first image here, this is the practice phase. Okay. Again, when the real test begins, you will not fall because you will be supported by the knowledge and practice. Okay. The first is knowledge here. This guy is practice, and this is you. Okay. So if you really want to flow across any difficult question, you must club knowledge with practice, and of course the support of Biotechnica. Okay, Biotechnica is always there for you, and we are starting a fresh batch from 1st of August 2019. Okay, last year our topper, you must have seen Ali Akhtar. She qualified 4 exams, my dear friends. She qualified with 5th rank in GATE, and she also got Inspire Fellowship. She also qualified ICMR, JRF. So there are various entrance exams she could qualify, but one can ask how. Because she asked the first question, if I have to get the topmost rank in CSIR net or GATE, then what else I should do? What all things I must have to qualify, right? And if we think that way, then we'll start arranging for the materials, arranging for the teachers who would support us. Okay, and that is why to all the students who join our coaching, we'll be giving free study material. Also, they'll be given with online study material which will have the video backup of every class. So you attend a class and then you can go back and see the recording of that class. So, and you can see unlimited times. So what happens is you have all that freedom to revise, re-revise, and actually go back to that actual class where they taught you that particular concept for which you're confused today. So this is how the video backup works. We also are providing the AIMGATE test series 24/7 chat support. Now how this works is the moment you have got a question, now generally most of you will study at what time? At night time, right? And that is where you will get doubts, and our experts will be online throughout the night to solve all your queries. We also provide you PowerPoints, animations, flashcards, quick notes for revision. You can ask unlimited questions to our teachers. So that is during the class and after the class, you can ask questions, and that is what science is all about. Science is the art of asking questions. There is no internal marks which will be deducted, so you should not be scared. Ask as many questions as you can, then only you can qualify CSIR, NET and GATE exam. You will also get a chance to interact with all our toppers, previous alumni of Biotechnica on the toppers platform. So this is all about the GATE course. Now, you must take charge of your destiny. Learning is earning, and today you're going to do that. Okay, in the next 1.5 to 2 hours, you are going to learn a lot, and that learning is going to translate into earning of the future. So pay attention, guys. I'll meet you at the end, and you must take control because something big, which is GATE 2020, is coming. Okay, so you must hire the right habits. You must hire the coaching of Biotechnica, you can say. You must fire all the bad habits like procrastinating, okay? Procrastinating, delaying, okay? And you must promote revision and practice. Then only you can qualify GATE with a high rank. Do not just target GATE exam. Target the highest rank possible in GATE exam, okay? Now, never give up. Like this tree, you can see it actually fell down, but still it is growing. Still it is growing. And that is what you have to do. Don't give up. Even if you fall, you may find some questions tough, some questions difficult. Even today, when our experts, I will pass on the controls, they will tell you certain things, you'll get scared, oh my god, how to do. Don't worry, okay? This is practice time. Okay, so don't worry at this time. You must never give up. You must keep practicing. Biotechnica is always around you on a digital screen near you. Since 2006, we are the most reliable brand in life sciences and chemical sciences today. I'm really glad to tell you we are on the top 40 websites in the world for biosciences. Okay, and that's a huge achievement for any Indian website. Okay, we get around 25-26 lakh scientists every day who visit the website and take notes or take information. So why you want to stay back? Biotechnica is always around. Learn from it. So today is the day when you will be listening to the eminent educationalists. Along with me, there are 5 to 6 more educationalists. You will be interacting with successful students who will be available in chat. You can ask your queries, my dear friends. All our experts are online in chat. Also, feel free to ask questions. Learn how you can enrich your career with add-on courses which Biotechnica provides. Again, I want to reiterate, failure is first attempt in learning, so don't be scared. Attempt that question, start practicing more, keep putting effort because effort never dies, and remember there is always a next opportunity awaiting for you if you don't give up. Okay, so that's all for now from my side, and now I will be passing on the controls to my fellow colleague who will be talking in detail about this basic questions and topics and maths and science, whatever, of GATE exam. Okay, so I hope you are enjoying the session right now. Shall we start with the real test, real workshop? Okay, now in the middle of the workshop, remember, if you get disconnected, just refresh and you will be connected back. That's the point number 1. Point number 2 is, if in case you miss this workshop, we will be only giving the recording of this workshop to those who attend the workshop till the end. Okay, the link to download the workshop recording will be given to only those. So make sure you are attending it till the end. Okay, and I'll be giving a surprise gift to all those who attend till the end. Okay, so that's a promise from my side. All the best to all of you. I am inviting my next expert, to talk to you. All right, are we ready? Yes, okay, perfect. So here we go, guys.
Hello everyone, I'm Dr. Tanushree and my talk is on General Aptitude and General Tips and Tricks for GATE 2020. You all are very much aware that GATE is one of the largest exams for PG courses in India. Every year, large number of candidates they appear for this GATE exam, but the pass percentage is very less. So you can imagine the level of preparation needed. Obviously, a lot of pressure will be on you, and that pressure is directly proportional to number of days left for the exam. So for the GATE exam, last moment study will not help. You have to start your preparation as early as possible. So going for any competitive exam, study cycle is very important. So if you all have this kind of study cycle, then it's quite good. But in most of the cases, it has been observed that you all end up doing this, right? So if you have the study cycle like this, it will surely not help. So what to prepare, how to prepare, and what type of questions asked are some of the questions that might be popping in your mind. So how do you get started? Everything is achievable by planning and management. So the first step is you have to be familiar with the exam pattern. Like what is the mode of exam, how many questions are asked, what are the total marks, type of questions, like they are MCQ, that is multiple choice questions, or they are numerical answer type questions. So for the GATE exam, you have both kind of questions, MCQ and numerical answer type. Then you should be aware that your paper is divided into how many sections. Then you should also be aware that whether there is any negative marking or there is no negative marking. So for the GATE exam, you have the negative marking. For MCQ, 1/3 for 1 mark question and 2/3 for 2 mark question. But for numerical answer type, there is no negative marking. General aptitude, as you all are aware, that it is common for all the subjects. You are giving exam from any discipline, you have to give this general aptitude as it is compulsory. So there are overall 10 questions that are being asked from this section, 5 questions of 1 mark and 5 questions of 2 mark each. So overall 15 mark questions will be there, and they are all multiple choice questions. Then second step is you have to go through the syllabus. You should be aware that what is included in your syllabus. So for the general aptitude, you have 2 parts. One is verbal ability and another is numerical ability. Verbal ability includes English grammar, sentence completion, verbal analogies, word groups, instructions, critical reasoning and verbal deduction. Whereas for numerical ability, you have numerical computation, numerical estimation, numerical reasoning and data interpretation. Verbal ability questions, they can be answered quickly as compared to numerical ability questions. Although numerical ability questions are easier than verbal, but they may consume time. So if you don't apply any shortcuts in computations, it will be very difficult for you to solve these questions in given time. So overall, this general aptitude section is very easy. You can easily secure around 13 to 14 marks out of maximum 15, and also the negative marking will be minimum from this section. So what are the books you need to refer? So for the quantitative aptitude, you can refer book by Arun Sharma. A modern approach to verbal and non-verbal reasoning, you can refer R.S. Agarwal. So basically, you can, for the verbal ability, you can go through class 10th English textbooks, and also you have to read the newspaper daily. And for numerical ability, as it focuses on approximation skill, so you need to increase your speed in calculations. You have to learn certain mathematical tricks for that. So for reasoning and aptitude, you can refer Naim Singh, and for high school English grammar, Wren and Martin is recommended. Next step is study smart and time management. To study, you have to set your goal, which is pragmatic and achievable. And you have to be extremely focused on your goals. So for GATE, you can assign units in a week. Okay, you can assign day slots per unit in a week. And also, you can assign hour slots for subunits in a day. You have to set a deadline for each section. Also, you have to adopt the approach which can help to better understand and remember concepts, so that at the time of exam, you can recall and apply them easily. Like most of the students, they follow rote memorization approach for preparing the exam. So it is okay, but it's not an accurate approach, because you should understand that this will not help, as simple memorization of topics, it may result in unanticipated errors. So you can learn by making flowcharts, diagrams, tables, okay. So you can analyze which approach suits you best and you can go for it. Similarly, time management is very important. Time management, like if your preparation is good, it will help you in reducing stress, right? And like it's quite obvious, nobody can study for longer periods of time with same concentration. So you have to analyze yourself. You have to work accordingly. Like you have to set your priorities, like which topics to be covered first, okay? Like some are good in doing calculations in early in the morning, and some find evening time best for learning or studying theoretical subject. So you have to analyze yourself and you have to work accordingly. Then it is important that you should also go through some questions from previous question papers. Because if you go through these questions from previous question papers, it will help you understand that what kind of questions are being asked. And most importantly, whatever you have studied the previous day, you should revise it the next day. So revision is equally important. Next step is evaluate yourself and practice. So how you can evaluate yourself? By mock tests. So these mock tests, they create an exam-like situation with similar pattern of questions and time limit to answer those correctly. Okay, so you should go for these mock tests. It will help you better understand your preparation and also help you locate your weaknesses. And next important step is practice. More you practice, better you will get at solving problems, okay, as well as time management, and there are lesser chances of mistake at the time of actual exam. So there are some other useful tips for you for preparation of this GATE 2020. You should be away from these distractions. Stay away from distractions. And what are these distractions? Your social media, your Facebook, Twitter. You just sit, you just think that, okay, I'll just give a reply of this message in a 2 minute, but that 2 minutes converts into 2 hours. So stay away from the distractions, and dedication, discipline and devotion. These 3 Ds are very important for giving any competitive exam like GATE. Next important thing is positive thinking, that yes, you can and you will do best in GATE exam. And the most important thing is that you need to stay healthy. Eat healthy so that your body, mind and soul is healthy and you can give your 100% in preparation for this GATE exam. Now there are certain useful tips for you just before a day of exam. And these tips are that whenever you are going for this GATE exam, just make sure that you have the print of GATE admit card, and you also check out the exam center a day before, so that you don't get panic during the day of exam. So you need to be aware of where is your exam center, how much far it is from your home, and how you can reach to your exam center. So these all are the things that you have to do before a day of exam. Then also you need to be well prepared for your exam kit. Like this is an online exam, but if you require anything, you need to make your exam kit. That your exam kit should have the admit card. If any photograph is being attached to it, you should have a photo ID. Okay. Then time management during exam. So the first important step is that you have to read the question carefully and understand it. Okay. Second step is that whatever the question is there, you have to analyze the right technique. Okay, so that you don't take longer time and you solve it in the given in a short or calculative time. Okay, so like if you think that this question is taking a lot of time, then just leave that question and move to another question. Okay, and whenever you are attempting the question, make sure that you save that questions every time before switching to another question. Next important point is that answer when you are completely sure. Okay, so this is just to make you understand that when you are giving any question, when you are giving the answer for any question, you can memorize the shortcuts or the diagrams that you have learned during the preparation of that question. Okay, and finally you should answer those questions only for which you are completely sure. So there are certain sample questions I have included in this session for you to make you more understand that what kind of questions are being asked. So like in verbal ability questions, you can have this kind of questions that choose the appropriate answer to complete the following sentence, that medicine is to illness as law is to. So 4 options will be given to you and you have to choose the correct one. Then numerical ability questions also, you have the question will be given to you and you have to solve and find the correct answer. So far whatever the tips and tricks I have told you, I hope that this will certainly help you, and you will, whenever you will be giving the exam, you will be like happy faces and not the sad faces. So in the end, I just want to conclude by saying a quote from Gail Devers, that keep your dreams alive. Understand, to achieve anything requires faith and believe in yourself, vision, hard work, determination and dedication. Remember, all things are possible for those who believe. So thank you students, thank you and all the best for the GATE 2020. Handing over the mic.
Good evening, everyone. I am Rashmi and we'll be continuing with the workshop for GATE BTA 2020 strategies, where I will pick up the portions from your Unit 1, Section 3, Section 4. So if you look at the topics that I'll be dealing with, it will be engineering mathematics, recombinant DNA technology, which is part of your molecular biology, all the techniques that it involves, as well as plant and animal biotechnology, including all the tissue cultures, etc. Right? So when you think about GATE, remember GATE comes only once a year, which makes it your priority. This is a chance that will stand before you only once. Every year you will get a chance, but remember, once if you are not able to give it, the next chance that you will get will be in the corresponding year. So of course I know all of you, none of you in fact, would want to waste your time, would want to waste that precious year, right? So with whatever time you have in hand, with whatever preparations you have in parallel, make sure to prioritize yourself. Because this is one exam that will help you not only in your enrollment for masters, it will also help you for your enrollment in, say, PhD, as a JRF, in various projects, and that will definitely be a beginning of a very bright career ahead. Now, while you sit for the exam, remember, time is like a pressure cooker. So, as we know, the tick-tock of the clock, the countdown, the timer, that definitely gives you palpitations, because that's how it works. It creates a pressure in your mind. But the more you have practiced, the more you are accustomed to the type of questions, the more confidence you will have during the exam. You will not feel that pressure, you will not feel that panic if you find familiar questions in front of you. And how do you do that? It all boils down to the practice that you do in the remaining time. So being a pressure cooker can either result in cooking you or producing something very delicious out of it. So it's all about the perception. It's all about looking at the struggle or looking at the end product. So remember, with every passing day, your practice must increase. The time you sit for studying must increase, and only then you will be able to cover the entire syllabus, so that at least when you sit for the exam, you do not feel trapped in a pressure cooker. Now, one of the key things to remember is, when you look at a question paper, read very carefully. There are different types of questions that can come in GATE. There will be some which do not have any negative marking. Yes, that's the beauty of it. It doesn't have any negative marking. These are called as the NAT or the numerical answer type questions. What are these? You will find certain questions which have a blank in it, which have a space provided at the end, wherein you are expected to fill a certain value. Now that value can be a decimal, it can be rounded off, the instructions usually are mentioned. However, the closest you are to the answer, the more accurate your answer is, the better you score in that. Hence, you cannot be scored negatively. Now, the other ones which are MCQ kinds, of course, there is a negative marking. For example, if a question is worth 1 mark, you deduct 1/3. If a question is worth 2 marks, you deduct 2/3. So depending upon the questions, you do not want to lose out on marks. You do not want your numerical type to go wrong. You don't want to skip the closest value. Hence, it is very essential for you to read very, very carefully. This is in terms of examination. Now, there's another reason you should read carefully, that is while you are studying. Yes, because if you look at an MCQ paper doesn't require too much of your analytical time. An MCQ paper will be very short and very direct and something out of the textbook that you have just left at home. So within these pages are your 100 questions that you are henceforth expecting. Now within these pages, if you do not scrutinize each and every term, you can leave those essential 1-2 mark questions, and that can be the reason for not qualifying in that exam. So number one, read carefully when you are practicing, when you are studying. Make sure to go line wise, make sure to remember those important key terms, remember those points, think about the mechanism. Don't blindly just engulf the text. Read, understand, memorize, retain it till your exam date. And how do you retain it? By revision and practice. Okay. So once you complete a chapter, it is essential for you to revise it every single day, every single hour, because till your exam is complete, that thing should be stuck in your mind. Correct? Now, the next important task for you is to assign weightage. Now, when I say weightage, every exam will have a different set of units. They will all be assigned a different number of questions, which is certain mark distribution. If you look at the GATE question paper, you have approximately 70% weightage given to proper subjective questions. Now within that 70%, there are so many units to be distributed, or within the 100%, there are so many aptitude portions to be distributed. So in order to understand that, it is important for you to assign the weightage yourself. For example, you can see portions from your general biotech, portions from your techniques, portions from your plant, from your biotechnological techniques, from fermenters. All of these, they are often asked in a huge number. Whereas if you consider the engineering mathematics, or if you look at the cDNA technology, or maybe if you look at these aspects or the animal or plant biotechnology, you may find it, yes, equally important, but possibly having a lesser weightage. So it is evident that I need to plan accordingly. How much time do I want to spend for how many marks assigned? Or how many hours shall I spend for how many different reference books that I have to cover? For example, if you look at Unit 2, that's the most vast topic which concerns about all the important reference books you can think of. Now to cover that, you will definitely require more amount of time, and you must also look at the kind of question that is coming from there. So how do you ascertain all of this? You can ascertain this by doing a 10-year paper analysis. So when you do a 10-year paper analysis, look into the type of questions asked per unit. Look into the marks and the weightage assigned per unit. Look for certain concepts that are repeated. No, questions will never be repeated, but concepts can be repeated. Numerical problems may be based on the same formula with a different twist. So this can only be done when you sit and analyze your 10-year question paper. Let's move into each section one by one. If I dive into engineering mathematics, there are different important formula-based areas that are very, very important. Example, matrices and determinants. Every question paper, one or the other question on matrix will definitely be there. Limit and continuity, that's another important thing. Sequences. Okay. Maxima, minima. When you go for limit, maxima, minima automatically follows. Laplace's equation, Laplace's transformation, another important thing. If you look at statistics, this is one of the most important thing, because as a biotechnologist, you must be strong in statistics. Mean, median, mode are the most common things, followed by standard deviation. You can have Poisson's variation, you can have binomial distributions because you have genetics involved. So all of these are definitely important. If I come to numerical methods, nonlinear linear algebraic equations, Simpson's rule, and then here and there certain differential equations. So if I covered these important topics mainly, I should be at least able to attempt a minimum of 5 to 7 questions from engineering mathematics. Now, the books that I must study. General aptitude can be picked up from your 11th and 12th mathematics. If you have your practice books left at home, you can definitely practice from there, because matrices, determinants, calculus, algebraic equations, these were definitely covered in your plus 2 courses. Apart from this, there are a lot of publications which come up with engineering mathematics general aptitude. So you can follow Arihant, you can follow Trishna. You can also follow these corresponding authors with different engineering mathematics and general aptitude books, which will definitely help you practice. Now in this area, practice is your key. These are some of the type questions: median, limit, matrix, etc. Come to recombinant DNA technology. Here, cDNA library, looking at RAPT, RFLP markers, site-directed mutagenesis, looking at southern and northern blotting, DNA labeling and sequencing, and identifying different various vectors and their restriction mapping can be proved to be important. These are some of the important places that you need to target. The books that you can use, for example, are, you can refer Gene Manipulation and Genomics by Primrose and Twyman. You can refer Molecular Biology of the Gene just to understand the basic concepts. You can refer books like Watson. You can refer books on genetic engineering. These will definitely help prune your concepts better in these areas. So understanding about the technique, remembering the concept on how to solve logically derived question, these will be important in this aspect. You can also get numerical type questions, for example, based on a molecular mass, based on a cDNA protein, based on the number of exons, introns, etc. You can also get MCQ type questions, which can be as long as combination questions, or they may be as direct as a single answer. Now, the last subunit that I'll be picking up for today is plant and animal biotech. This is a very direct topic. A direct topic in the sense, if I just remember key concepts involved in animal cell culture mainly, and certain metabolites involved in plant tissue culture, these are the areas along with hybridoma technology, stem cell technology, which are most important in this unit. So along with this, you can always look at the growth kinetics, you can always look at the culture media, conditions, etc. But the most important thing is looking at the techniques, looking at both PTC and ATC, looking at the derivative techniques like hybridoma technology, how it is put into use, because this is all about biotechnology. The books that you can follow for biotech is, there are many authors on this. You have publications from Oxford and Springer as well. You have the most famous B.D. Singh and the Verma and Singh. So these are the key biotechnology books that one can use. Apart from this, to clear your concept, basic physiology books can be used. However, you have to focus on the experimental aspect more. The type questions can again range from direct questions to combination questions, wherein you have to understand the mechanisms, each and every component, how it functions, specific nomenclatures, etc. If you do this, these are direct 1 or 2 mark questions and they are very simple for you to answer. Now with that, the only point that I would like to emphasize is revise. How do you do that? Because these are key concepts that you have been studying since your UG and PG. You have to revise PG for some. You must revise this because it's very easy to forget, but it's very difficult to retain it. How do you do this? By sitting for different mock tests. A mock test is a very good analysis of your progress and it will give you the exact limitations of your practice. So revise well, study well, utilize your time, practice however you can, and all the very best for your upcoming GATE 2020 exam. Thank you.
Hello everyone, I'm Dr. Sunita and today we are going to discuss about the strategies for GATE 2020. Now before we begin about these strategies, let us try to understand what this GATE is. It stands for Graduate Aptitude Test in Engineering. So is it only an engineering exam? Actually, GATE is a national level exam, entrance exam, which is conducted every year for different subjects. It's not only engineering subjects. In fact, the test is also for chemistry, for physics, mathematics, and life sciences as well as biotechnology. So today, mainly, I'm going to focus on biotechnology. So if you look into the syllabus of the biotechnology, it has been divided into different sections. So section 1 is engineering mathematics. Yes, there is some amount of mathematics. Then section 2, general biotechnology. Section 3 is recombinant DNA technology. Section 4, plant and animal biotechnology. Section 5, bioprocess engineering and process biotechnology. So these are the 5 sections that this entire syllabus is divided into. So I will be mainly focusing on the section 2 and the section 5. So let us first try to understand how this exam is conducted. Of course, it's an online exam. I guess all of you know it very well. And the duration will be approximately 3 hours. Okay. Now, if we try to understand the marks weightage, okay, for whatever syllabus we saw. So, majority of the marks is assigned to the subject-related questions, 70% of the marks. And engineering maths is 15%. And general aptitude is again 15%. Okay, so this is how the marks is distributed. Now, pattern of question is, of course, it is total 100 marks paper. Duration, as I already said, is 3 hours. Total question will be 65. And yes, we have 2 basic types of question. One is multiple choice. This is either 1 mark or 2 mark. So some questions of this type will be of 1 mark and some questions will be of 2 mark, and there is a negative marking. So for 1 mark question, negative marking will be 0.33 and for 2 mark question it will be 0.66. Whereas there is also a numerical answer type question where there will be some problems given. You have to do some mathematical calculation and then you have to just write the value. So here you don't have any options. In MCQ you get options, but here you don't get any option. So in this case, one good thing is there is no negative marking. Some questions you will find will be little bit big questions. They will be, having some paragraphs. So you have to comprehend the paragraph and then you have to answer. So both in MCQ and NET you will find that. As well as such type of questions are often related. That means you will have maybe question number 45. So you have a paragraph. And then you have question number 45 and you have question number 46. So both are related to the same the paragraph. So what is important is you need to comprehend it and then you have to answer the subsequent question. So this is the pattern of question which will be asked. But remember, negative marking is only for MCQ, not for the NAT. But NAT you need to do the calculation. So that is the basic way the exam is conducted and the questions are distributed. So we saw the general syllabus. We also saw that how the exam is conducted and what is the pattern. Now let us try to understand how to prepare. What should be the approach towards preparation? So there are 3 important steps. First is planning, then preparation according to the plan, and the last one is the performance. So if you do these 2 very well, of course your performance is going to be the best. So when I say planning, the first thing that you need to do is read the syllabus. You need to find out what all is there in the syllabus, and after that what you need to do is you need to identify already what all you know. So maybe you have some specialization, so those topics you already know. Some topics you know but maybe very little, so you have to choose those out and accordingly you have to make a timetable. So you should know, okay, this to this time I will study this, maybe I will revise it every week. So all these plans you should be making, that is, should be reflecting the timetable. And often what do we find is, after making timetable, we find it very difficult to continue to follow the timetable. So it is better that you set up some short-term goals, okay, and you try to evaluate yourself time to time. Okay, so maybe you plan that within this 7 days I will finish maybe section 2 and then I will do a revision on the 4th week. So like this you try to set some small goals. So this is what comes in planning. Next, preparation. So most important thing is you need to have a reference book and you need to have the previous year question papers. Reference book will help you to get the knowledge, okay, and question paper will help you to understand what type of questions are asked, which portion actually you need to focus more. So best is when you do your studies, rather than maybe writing illustrated notes, you try to make flowcharts. You made some diagrams. That way you can better remember the particular topic. You can better understand it. In case some concepts you are finding very difficult to understand from reading, then it is more preferred that you search for videos or animations on that particular topic. Then if you visualize it, it becomes very easy for you to understand the concept. So this is the way you should be preparing, and of course you should go on solving papers. Finally comes the performance. So of course when you do your planning and your preparation well, what do we find is performance is definitely going to improve. Okay, but during giving the exam, the final exam, try to be in a relaxed state of mind. Avoid any last minute hurry. When you read questions, read them thoroughly. This is more important when you are solving those paragraph related questions, okay? So try to read the question, comprehend the question in a very cool mind, and when you find related question, try to understand how one question is actually different and what exactly that question is asking. Try to avoid negative marking and of course try to have a good time management. So it is better to first go for the MCQs and don't waste time. Whatever comes in your mind, you try to mark that as an option. Then once you are done with MCQ, you can go for those comprehensive type questions, related questions, as well as the numerical type questions. So better you plan that what way you will begin, how you will proceed, and try to mark whatever is coming as a sure answer, sure option for you. Okay, don't wait, because it will again make you to lose time, and maybe unnecessary thinking will lead you to a wrong option. So again, negative marking. So try to follow all this, then definitely you will be able to give a better performance. So as I said, I'll be discussing about the general biotechnology and the bioprocess engineering. So this general biotechnology is again divided into biochemistry, microbiology, cell biology, molecular biology and genetics, analytical techniques, immunology, and the bioinformatics. In biochemistry, mainly the structure, function of biomolecules, biological membrane, okay, the action potential, transport, okay, these questions may come from this portion. Enzymes are also very important, enzyme kinetics, then how exactly inhibitors are working, so questions are asked from here. Then of course the metabolism, so how the carbohydrate, lipid, amino acid, mainly bioenergetics is focused on. Okay, how many ATP, how many NADPH. So these type of questions you can get from this particular portion. Now for preparation, as I said, you need a good reference book. So the reference book which you can follow here is, of course, Lellinger, Principles of Biochemistry, then Biochemistry by Berg and Lewitz-Steyr, and next is Biochemistry by White and White. Okay, these are the very good reference books that you can follow. Next, microbiology. So mostly classifications of the microbes, okay, viruses, and what type of methods is done for the growth, aerobic, anaerobic respiration, host pathogen interaction. So you will find many questions based on diseases and their causative agents. So match the following, those type of questions. Basis of growth curve. So those type of questions you can expect from this particular portion. Again, the good reference book for this particular topic is, of course, Microbiology by Prescott and Microbiology by Tortora, Brock Biology of Microorganisms by Madigan Michael T. So these are the very good books. If you can use them, you will find that most of the whatever given in the unit in this particular portion will be covered by these books. Next is cell biology. Cell biology, basic structure of prokaryote and eukaryote, basis of cell cycle. So many questions will come based on the mitosis, meiosis, how many cells, how many chromosomes, those type of questions. Then of course cell signaling, communication. Okay, these signaling questions are also very common from this particular portion. Reference books for this topic is Molecular Biology of Cell by Bruce Alberts, then The Cell, A Molecular Approach by Cooper, Molecular Cell Biology by Lodish, Cell Biology by Karp. So these books are sufficient to cover up this entire topic. Next we have molecular biology and genetics. So basic structure of genes, chromosomes, then mutation, of course replication, transcription and translation and their regulation. You can find questions, mainly replication, many question comes. Then Mendelian inheritance, okay, the probability of getting one particular type of the phenotype, linkage, recombination, then mapping, many questions are asked from mapping, okay. Then extra chromosomal inheritance, horizontal gene transfer and transposable elements also question to come. DNA damage and repair, molecular basis of genetic diseases. Again you can get some match the following question from here. Reference books, Principles of Genetics by Garner, iGenetics, A Molecular Approach by Russell. So these two books are very good books for this particular topic. Next is analytical techniques. So different types of microscopy, spectroscopy, chromatography, electrophoresis, all this are covered under this topic. You can get questions based on electrophoresis, maybe based on the microscopy. Chromatography questions are also asked. So basically techniques or technical question based on this particular topic, they can be asking. They can give you some experiment and they can ask you, okay, if you are suppose isolating two proteins using a ion exchange. So based on the property of protein, how they will be eluted. So these type of question you can expect. To prepare this particular topic, you can use these books, Principles and Techniques of Biochemistry and Molecular Biology by Wilson Walker, Introduction to Spectroscopy by Pavia, Principles of Proteomics by Twyman, and the Introduction to Microscopy by Suzanne. These are some of the books. Next is immunology, a very vast topic, but try to focus on innate, humoral immunity, differences, the antibody, structure, function, types of antibody. Then try to focus on the T and B cell, how they're getting activated, what are the receptors, MHC, antigen presentation, and many questions are asked from graft versus host reaction. In fact, sometimes you can get questions on hypersensitivity, autoimmune disorders, mostly match the following type of questions. So reference books is Essential of Immunology by Reut or Immunology by Hubey. These are very good books, enough for covering this particular topic. Lastly, under this section comes the bioinformatics. So knowing these softwares, the tools is very important. So tools for data mining, the softwares for like data mining, molecular dynamics, simulation, okay. How to do the scoring, okay, what type of software can be used for the sequence alignment, what are the different databases. So all this are very important. Books is Introduction to Bioinformatics by Arthur Lesk and Essential Bioinformatics by Jim Beyond. These are very good books which you can follow, basic books. Next we have Bioprocess Engineering and Process Biotechnology. Now in biotechnology many questions are asked from this particular portion. It's mainly about the reactor, maybe scale up, how the reactor design is done, rheology, the heat transfer, mass transfer. So questions, many numerical questions are asked based on this. Aeration, agitation. Then we can also have questions from kinetics, that is microbial growth, how the substrate utilization is taking place, product formation. So problems can be asked based on this. Problems based on scale up can also be asked. Apart from that, question may be asked about the different types of fermentation, batch, fed batch, continuous. Then what way the separation can be done, like solid-liquid separation, liquid-liquid extraction, okay. So these are one of the most important topics based on which questions can come, and mostly what type of questions are coming? Problem-based questions are coming, okay. Apart from that, okay, downstream processing, process design and development, okay. So again, this is the scale up. You can also be getting questions on the different type of cell cultures and biofill, bioplastic, industrial enzymes. Here, immobilization of biocatalysts. From this also, many problems are asked. So numerical problems may be asked from this particular portion. Now, reference book for this particular topic is Bioprocess Engineering, Principles by Doran, Bioprocess Engineering by Schaller, Protein Purification by Scopes, Principle of Proteomics by Twyman, Principles and Techniques of Biochemistry and Molecular Biology by Wilson Walker. These are the books that can be used for this particular topic. So try to focus on these aspects, whatever I have said, and also you can check the previous year question papers to find out which portion is more focused, and based on that you can prepare. Try to develop the concepts, and better way is you draw flowcharts and diagrams for that. And of course, often give exam. Mock test, try to give more mock tests so that you can manage the time better. So that's all for the workshop session. Thank you all.
Hello everyone, myself Dr. Preeti Saini, and as suggested previously, I am going to give you certain suggestions and tips regarding XL or Life Sciences portion of GATE along with Dr. Priya Ved. I will be discussing what are the important portions, what are the topics those should be covered, and what kind of questions are expected in GATE by looking at previous year GATE Excel question paper. Okay, so we are going to discuss certain questions as well, and the topic which will be covered in this 15 minutes will include biochemistry, microbiology and zoology. So by now you have got a lot of tips and tricks in general regarding any MCQ exam, how to prepare yourself, how to prepare for general aptitude, and I am not going to repeat the same. So let us directly jump into biochemistry preparation strategies. Now, pH buffer, this is a very important part of GATE. Every year you get 1 to 2 questions from this topic. Now for all of us who are not from chemical science or chemistry background, we find it difficult to even check the log and anti-log. But that is important. Checking log, anti-log forms not only the part of pH buffer but also thermodynamics. So you need to learn that thing clearly. You need to differentiate between natural log, log 10, what are the important buffers, what are strong acids, strong bases, how they differ with weak acids, weak bases. Mostly numerical part is involved here, and pH buffer type of questions along with thermodynamics, enzymes, proteins are covered not only in GATE but also in entrance exams as well as CSIR. Okay, so you can learn the theory part from Voigt and Voigt or Leninger as well. However, if you want to solve the numericals, please try to look for more than one source. For example, you can go through various websites, question papers, books, okay, and previous year CSIR GATE question papers also. That will help you to get a hold on the subject. Thereafter, amino acids and proteins really important. Okay, every year 4 to 5 questions are expected. In fact, their interaction with DNA, the carbohydrates, lipids, all the biomolecules are quite important. You can see proteins or amino acids are more important with more weightage, 4 to 5 questions every time. However, carbohydrates, lipids and nucleic acids together give 2 to 3 questions every year. But what should exactly be studied? As you know, Ramachandran plot, pK values, charges on proteins and peptides, secondary, tertiary, quaternary structures, hemoglobin, all of these are quite important. Similarly, in regard to carbohydrates, what you require is, you require to know stereoisomerism, glycolysis, metabolism part, along with linkages, alpha 1,4, beta 1,4. What is the difference between these two? You should know, right? And main concern should be at ATP generating sources. Also, it is very necessary when it comes to carbohydrates that you should know all the steps involved. What is important of those steps? When it is occurring, where it is occurring, in which part of the cell it is occurring, in mitochondrion or in cytoplasm. Okay. For nucleic acid, sequencing is important. Sequencing is also important from protein part. Metabolic pathways for nucleotide synthesis. Replication, it still holds important. Lipids. Most important thing is lipid bilayer structure. Okay, followed by the structure of different types of lipids and different types of nucleic acids. Now, by nucleic acids, I mean nucleotides A, C, T and G. Fatty acid metabolism, which involves phospholipases, different types of phospholipases. What are the differences between them? Where phospholipase A works, where phospholipase B works. Saturated, unsaturated fatty acids, their receptors. Permeability of lipid bilayer. How melting points changes with structure of fatty acid and how it affects its mobility as well. So all of these things are important and should be considered well. That is why we have these standard reference books, Stryer, White and White, Blenninger, to go across these biomolecules, their structures, functions, metabolism, as well as composition. So biochemistry, most of us, we consider it to be a very quite difficult, right? Especially for those of us who have not studied any type of biochemistry in post-graduation or graduation levels. So I am going to tell you what are the most important topics and how you can prepare them and what are the relevant books. So considering that chromatography is one of those units which is of utmost importance. Every year at least one question you get directly or indirectly from chromatography. Basically, the questions are based on size exclusion and ionic type of chromatographies. You know size exclusion, which gives the separation based on sizes, and ionic, which gives the separation based on charge interactions. So these two you should remember and study in detail. You should know their principles. For example, if you consider size exclusion type of chromatography, you have been given suppose 4 different sized molecules. Out of those, which of the molecule will be eluting out first? What is the volume of elution buffer required here? All these things you should know in detail. Also, there are certain formulas regarding wide volume, VE. All these you need to remember. So for preparing chromatography, you can always refer to Principles and Techniques of Biochemistry and Molecular Biology by Wilson and Walker. Even there are certain editions which are available free of cost online that you can refer to. Okay, so this is of utmost importance. After that, protein. Protein forms an important aspect in any exam as far as biochemistry is concerned. Okay, even if you want to understand enzymes, you need to go for proteins first. It is very important, and every year you expect 1 to 2 questions out of protein metabolism itself. And which points you should focus on? Those include urea cycle, the enzymes related to urea cycle, where exactly urea cycle takes place, which ammonium from what source is actually forming urea, the synthesis of amino acids, and the related diseases. All of these should be on your tips. And as all of us we know, Leininger and Word are the holy grails for proteins or biochemistry in general. Other than that, you can also refer to Stryer if you find that easier to understand. As I said, enzymes related to proteins once again, quite important. Every year you get 2 to 5 questions from enzymes, directly or indirectly. It may be match the following type of question. It may be a question related to enzymes from certain cycles. It may be related to Michaelis-Menten equation, like Lineweaver-Burke plot, Hill coefficient, or inhibitors. Inhibitors are quite important. These are difficult as well, but if you get proper guidance, you will come to know how to remember, what to remember, and how to solve the questions which are related. So, numericals are always expected and should be done perfectly, which means every day you should take 5 to 10 minutes out of your schedule to practice certain questions from various sites, various books, various question papers regarding enzymes. Enzyme is not only important from GATE point of view, even CSIR, entrance exams for PhD, they also consider enzyme as the important part. Okay, so again, the theory part you can get through Leninger, but for numericals, please try to look for more and more numericals through MCQ books or previous year question papers. Maybe standard reference books as well. Also, you can refer to NCBI site, which has certain standard examples of enzymes. Immunology. Now, immunology is important from biochemistry, zoology, even microbiology point of view. So, QP's immunology is the standard book which everybody recommends. Quite important, every year you get 1 to 2 questions in case of biochemistry part. However, you also get questions from immunology in zoology and microbiology. So, it is quite important. In fact, we will see later on that in zoology and microbiology as well, there are various questions from biochemistry portion. So if you are clear with biochemistry, it will be easier for you to perform in zoology and microbiology papers as well. Okay, so learn in details B and T cells, their functions, hybridomer technology and how it can be implemented. Serological test, precipitation, agglutination, complement fixation, fluorescent antibody technique. Okay, so these are of most importance, but you should go through the book thoroughly. Okay. Then rDNA technology. Now rDNA technology is the whole and soul of biotechnology. So you should learn the details and basics of everything related to rDNA technology. It is very important. Every year you get 2 through 3 direct or indirectly related questions related to gene cloning or rDNA technology. Learn Western blot, Southern blot, phage, electrophoresis, their applications, and if there is some situation like they tell you this is a protein, how it has to be separated, you should know how out of Southern blot, gel electrophoresis or Northern blot or Western blot, which is the one which is most applicable in that particular situation. So interpretation and molecular weight analysis of the given gel, this is also required. Vectors, their carrying capacities you should know. Like you should know human based vectors have maximum carrying capacity compared to yaks or bats or any other artificial chromosome. Learn the genetic code and its implementations as well. Again, you can refer to Wilson and Walker. Thermodynamics. Now, thermodynamics is also going to give you goosebumps in zoology portion. So, thermodynamics, I know, being the students of science or biological science, you find it quite difficult to deal with thermodynamics or physical portion. But this is important. Please refer to the theory portion from Leninger and try to solve maximum questions from Gibbs free energy, standard free energy. You should know the difference between these two, okay? And how these are related. Enthalpy, entropy, work, internal energy. These are at least the basic ones which you should be knowing. The related examples, the R factor, how temperature can be converted from Celsius to Kelvin. All these things you should know. You should know the value of Faraday constant, how there are different values of gas constant, why are there different values of gas constant, how you should implement them to find out free energy. Okay, all of these are quite important. Then comes microbiology. Now in biochemistry I have tried to tell you most important parts, but yes, other parts also you can try if you have sufficient time. For example, if you start preparing now onwards, you are having plenty of time to actually go through all the topics. Here the important topics include antibiotic based questions. Which are the antibiotics that inhibit DNA synthesis or RNA synthesis or protein synthesis, or they affect the functioning of protein, which are the competitive, non-competitive inhibitors. Those can also be a part of antibiotic. Preservation, sterilization techniques like autoclaving, their principles. Bacterial physiology and morphology like cell wall, cell membrane, how penicillin and lysozymes affect synthesis or they cause degradation of bacterial cell wall. All these things are important. Growth cycle, lag, log, exponential, these are important. Questions based on them, numericals based on them, those are important. Remember, log and not by and t by and not. Okay. Then disease based question. I always tell you that diseases are of quite importance. You should know everything about them. Which microorganism is causing what disease? Which is the particular medium for that particular microorganism? Conjugation, transduction, transformation, that is DNA transfer among the microorganisms. This same thing based on immune assays. Again, it is important. Viruses, the diseases caused by viruses. Carrying capacity of DNA viruses as plasmid. Which virus affects animal, which virus affects plants. All of these things are important. Vaccines. Then fermentation. Fermentation you can always read in detail from this particular book, Stanbury Whittaker and Hall, which is even available online free of cost. So this is going to help you solve difficult questions. Okay. Then whatever you may think about history part, but that is important. Like you see this question where you have to answer based on the scientists and their major contributions. This may seem quite boring to you, but it is going to help you score something, right? Similarly, microscopy. You are reading microbiology, that means microscopy has to be studied in detail. Whether it is electron microscopy, confocal microscopy, atomic force microscopy, light microscopy, dark field microscopy, anything related to microbiology. Again, you can make a table to differentiate between all of them, right? Like I said, culture media are important. This is one of those questions. Complex media, selective media, how you define them? Here, option B is correct. Then, zoology. Now we are short of time, otherwise I would have discussed more questions. So if you are quite interested in taking up this exam and getting it clear, I would suggest you to refer Biotechnica. You can purchase the study material as well. Okay. And for GATE aspirants, we have special online coaching as well, where we will be discussing these type of questions, how to solve them, what are the important books, right? Like for example, in case of microbiology, Pelzar or Prescott are always needed. For diseases, media-based questions, you have Ananta Narayan and Panikkar as the holy grail. Even if you get last or previous editions, go for them. Okay. Similarly, zoology. In case of GATE Excel zoology, the important topics, most important topics are physiology and metabolism from heart, kidneys, lungs. You will get different sort of small and large questions. Okay. Even if you don't know the excretion part, that will do. But go through heart, kidney and lungs of different organisms. How they differ from each other. Which organism has 3-chambered heart? Which has 4-chambered heart? How their functioning varies. Diseases, again, parasitic diseases are of quite importance. Diseases, symptoms, treatments, okay. Classification of organisms. Speciation and selection, which is actually a part of ecology, that is also covered here. Immunology, once again. Genetics is also covered under zoology. Enzymology, once again. Cell structure and function. So basically for zoology you can refer to Tortora and Derrickson, which is Principles of Anatomy and Physiology. But yes, if you have other questions, other issues, other problems related to immunology, you will have to go for QB. For genetics, you can go to Snowstad and Simmons. For developmental biology, you can always refer to Gilbert. Then cancer is of quite importance. Again, you can see DNA isolation, chromatography, PCR, electrophoresis. There are a lot of topics which are covered under zoology itself. Cell cycle, blood group, Hardy-Weinberg equilibrium or replication itself. Till date you are getting questions from replication, glycolysis, basic things. So try to clear your basics, only then it will be possible for you to solve the detailed portions. So I hope I have told you the important points, but has to be remembered, how you can remember them. And here my part ends. I would like to hand it over to Dr. Priya Beth, who is going to help you in further regards regarding Excel paper of GATE. If you have any more questions, you can write back to us. Okay, thank you so much.
Hello students, I'm Priya Bhatt and thank you Dr. Preeti for talking about preparation strategies of biochemistry and SYLLABUS. Okay. And now I'll be covering some of the preparation strategy of chemistry, botany and food technology of the life sciences portion of GATE. Okay, so here again I'll be dealing with the important topics for the exam point of view which you need to study. Okay, and as you know the paper of GATE, if you see the previous year question paper, which is generally MCQ based, and some question come maybe in the fill in the blanks or something like this, but overall it is MCQ based. Okay, so accordingly you need to prepare. Okay, and we will be seeing about the important topics of chemistry, botany and food technology. So first I'll start with the important topics of chemistry, then botany and at the end food technology. So seeing about some of the chemistry topics, first one is the atomic structure and periodicity. Generally 2 to 3 questions come in exam, and again it varies from year to year. And the important topics which you need to cover in this is the Planck's quantum theory, which is very important. Then wave particle duality, like a light has the both nature, wave nature as well as the particle nature, and all the equations related with this concept that you need to know. Then your electronic configuration of atom, like what are the orbitals are there in the atom, starting from where the proton, neutrons and are there. Then from that particular part you need to say like where your electrons do move. All this thing you need to know over here. Then periodic table is really important. Though we studied periodic table during our school days, maybe in 9th or 10th you might have studied. So it is very easy, but you need to know about the periodic table. Why it is important to know about periodic table? Because if you know about periodic table, then it's very important you to know about ionization energy and the electronegativity stuff. Okay, so which atom has which high electronic which atom has high electronic negativity, which have high ionization energy. Likewise, you need to know about this one. Okay. Next is structure and bonding. So in the structure and bonding, ionic and covalent bondings are important. So ionic means it's kind of a salt bridges are made. It's an ionic bonding which is a positive charge and the negative charge, and that's a kind of ionic bonding. And in the covalent bonding, generally electrons are shared. Okay, so again this is very important. And Vesper theory is there. You need to know about the shape of the molecule. Hybridization, resonance. Resonance is the concept where when two atoms are bonded by double bond or triple bond. So one bond may be partially double bond or single bond. So it gives you the concept of resonance. Okay. Then structure parameters. Again it is important. The bond length, bond angle, bond energy. Again, these factors do vary depending on the type of bond which is there. If it is a double bond, its length will be different. If it is a single bond, its length will be different. All those things you need to know. then hydrogen bonding, van der Waals interaction, ionic solids, ionic radii. So basically here we are studying the bonding pattern and the structure of atom and molecule. Again you need to study about this thing. And questions generally come from this part is 1 to 2. Then S, P and D block element, as I've told you, you need to study about the periodic tables. So once you know the periodic table, you will be knowing which atoms are falling into S block, which are falling into P, and so for the D block. And accordingly you need to study about their oxide, halides, hydrides of these elements. And yeah, definitely you need to study about the coordination complex and the valence theory. Because valence bond theory is very, very important while you are studying the coordination complexes. So you might expect 1 or 2 questions. Again, sometimes it may not come in a particular year, but it is important for you guys to know. Then in the chemical equilibration, so in the chemical equilibration, we need to study about equilibration effect. We need to study about the pH and buffer effect. Okay, and what is the equilibrium constant? These are the main important things for you guys to know in this particular topic. And again, this sometimes 0, sometimes 1 questions come from this. Okay. Whatever I have discussed so far is part from the physical and the inorganic chemistry. So in the chemistry, you basically need to cover all the 3 chemistry: inorganic chemistry, physical chemistry and the organic chemistry. These 3 have their own equal weightage. Okay. So, and again, chemistry is a compulsory subject. You can't opt this out. So, your preparation for chemistry has to be really strong. Okay. Then talking about the electrochemistry, not many questions come from this part. Okay, you might expect 0 or 1 question. So it will be talking about the electrical conductivity, Nernst equation, which is important, Nernst equation. Then yeah, something about the thermodynamics again is important. But for thermodynamics, again there is a full different unit is there. So you can cover thermodynamics over that particular part. The next important thing is about the reaction kinetics, where you will be studying about the rate of reaction. You will be studying about the first order, second order, 0 order reaction and rate constants. Okay, again these are very important. And here you can study about the role of enzymes in changing the rate reaction, and accordingly your rate order may get changed. Okay. So all these things are important over here. Then yeah, as I told you, for thermodynamics there is a separate unit. So in the thermodynamics, what is important? All the laws of thermodynamics, that is first, second and third law. Then you need to study about enthalpy is there, then entropy is there, then free energy is there, Gibbs free energy. All these are very, very important for you guys to know how the equation and the other stuff which is related to all this. Then talking about the organic chemistry. So in the organic chemistry, many things you need to know. You need to know about all the organic molecules, what kind of reaction they form, okay, and how you can identify the functional group, and if a functional group is attached to an organic molecule, what is the significance and what kind of reaction they can carry out. All this thing you need to know in the organic chemistry. And elementary treatment is like S1 reaction, SN2 reaction. All these things you need to know. And identification of functional group by chemical test. So when you will be studying through the book, you can have a glimpse of this. And last in the chemistry part is super reactivity correlation. So here we study something about what is acid, what is base, all this thing. Then how different definition of acid and base. Okay, and you need to study about the isomers, optical and geometrical isomers, then tautomers, conformers. Okay, all this correlation parameters with respect to a structure of a molecule, you need to study over here. Again, you might expect 1 or 2 at least question from this particular part. Okay, so this is about chemistry. Now we will be seeing something about what topics you need to cover in the botany. And yeah, before going to the botany, so these are the books for chemistry you can refer to study more about this. So this is by Carey, Organic Chemistry. For an organic chemistry, you can go for a book by K.D. Sharma or Jugiri Paula. And for physical chemistry, in organic chemistry, this book, Donald Data. This is also a good book. And for physical chemistry, you can go for this book. And if you have your own good book like which you studied during your maybe bachelor's, even you can refer those books as well. But these are kind of standard ones. It's better to you can refer these books. Now moving ahead with the botany. So first part in the botany is the plant systematics, which is mainly related with the taxonomy of the plants, the classification of the plants and the plant grouping. So generally the question doesn't come from this part, but you may sometimes expect one question coming from this. Now next is the plant anatomy. So in the plant anatomy, you need to know about the structure, what are the organs is there, what is their structure, cell wall structure, and xylem, phloem. This is basically a vascular system is there. Then secondary growth in the plant, like adventitious root is forming, how they do form. Okay, all this stuff you need to know in this plant anatomy part. Then in the morphogenesis and the development, it is mainly about your developmental biology in the plants. So here you need to know from the gamete formation till the full plant how it is being generated. So gamete means your pollens and eggs are how they are being formed. So how cell cycle, cell division will take place. That is, you need to know about the meiosis and mitosis over there. Then how the fertilization is taking place. After fertilization, embryogenesis, seed formation, and then how the plant get formed, how the seed get germinate, and how the seed maintains their dormancy. Okay, all this thing you need to study over here. And other than this, something of the plant biotic part is also being covered over here, related with the clonal propagation, soma clonal variation, embryo culture. All this part comes under the plant biotechnology, in vitro fertilization, artificial seed. So you need to study a bit about these stuff as well. You may expect one or two questions coming from this part. Sometimes it may be 3 as well. So be prepared for this. Then next is physiology and the biochemistry. So biochemistry part, like general biochemistry part, as Preeti Ma'am has already discussed with you, all this thing you can cover. But other than this, only with respect to the plant physiology you need to study, like what is the plant-water relationship, what is the role of plant hormones, what is the role of photosynthesis, what is the role of this stromal physiology, signal transduction in the plant, and transport of solids and minerals under the stress condition. Then flowering in the plant is again important part over here, that is photoperiodicity. Okay. All these are very, very important things for you guys to remember. And yeah, you might expect at least 2 or 3 questions from this part coming. Okay. Then moving to the next topic. Next is again genetics. So about genetics, it has already been told to you. So again, the normal genetics like Mendelian inheritance, extension of Mendelian inheritance, what are the gene mutations are there, what are the type of chromosome mutations are there, linkage, recombination, all these things you can study in the genetics as a general stuff. And you question actually come from this part in the botany section. You may expect at least minimum 2 questions always come in paper which I have seen. So you can go through the genetics portion. Then the next is the plant breeding and the genetic modification. So here you need to know about some of the stuff of doing the plant breeding, hybridization of the plant. Then how you can create haploid plant, triploid plant, or multiploidy plant. Okay. Then how you can form transgenic plant using some markers like herbicide resistant plant, then antibiotic resistant plant, stress resistant plant, drought resistant plant. Okay, so how you can do this thing by genetic modification, that you need to study. And question may come from this part, like at least 1 or 2 questions you may expect coming from this part. So be prepared for this. Then the next is economic botany and ecology and environment. So in the ecology, economical botany, mainly you need to know about those plants which are of economical use or the medicinal use, like the cinchona from which malaria medicine has been formed. Like this, you need to know, because question may come from this, but at least sometime 1 or you may expect 1 or 2 questions coming from this part. The economic importance of all the microflora like algae, fungi, lichens and bacteria. Again, this is very important. Then next is ecology and environment, in which you mainly need to study about ecosystem, food chains and cryopreservation, etc. Generally, questions doesn't come from this part, but sometimes you may expect. The last is about the plant pathology. So in the plant pathology, you need to study about the classification of plant disease, like the important pathogen and how you can detect those pathogen and how you can control their measure. That you need to study. And again, plant microbe interaction is again important, which you can study over here. And question you may expect from this part coming minimum 1 or 2. So not more than that generally come. So this is about the topics which you need to study for the botany, and these are some books which you can refer. The book by the Plant Pathology by Pandey, then Plant Physiology book by Jajier, then Botany book is there by Singh and Pandey. This is really good book. I've heard about this book, and for genetics you can consider any standard book. Pierce, Gardner is there. So any standard book you can consider for the genetics. Now moving to the next and the last part, that is food technology. The topics which you need to cover in the food technology. So first is food chemistry and the nutrition. It is very same as that of your biochemistry, where you study about all the macromolecules. Here you need to study about the carbohydrate, the structure, classification, what are the type of carbohydrate, all this thing and the function. Okay, so for as well as for protein, lipids and so, you need to study about the pigments, like which type of pigments are being there in the food, some like chlorophyll is there in some grain food, like this, xanthan is there. And there are, you need to study about the food flavors, the plant compound which are giving a food a particular flavor. That all the things. And enzymes are very important to study. Then in the nutrition you can study about the vitamins which are very important, minerals which has to be there, and the role of essential amino acid in the food. Okay, like if I'm talking about a vegetable, so what is the content of vitamin in that vegetable, nutrient, the content of fatty acid, content of amino acid. All these things are very important to study. So, and again you can expect 3 to 4 questions coming from this part. Then food products technology. So here this part basically discuss how when a food processing is being done in the marketed food, how it is being done. So if I am talking about a liquid food, so for example milk, so it has been properly pasteurized and pasteurized, and then it has to be packed in the tetra packs. Okay, so again there are layerings in the tetra packs, and how you can process each and every food, okay, before sending them to the market so that consumers can use this thing. You need to study about all this thing, how what happens for each and every food product. Okay. Like for grains is there, fruits and vegetables is there, for animal products. For each and every type of food product, we need to take different precautions while packing, and there are different types of packing are there, and the processings are different. So it's not like you are studying for one and you can go for the others. So you need to study processing for each and every type of food product separately. Okay. Next is about the food microbiology. So here mainly we are studying about the type of microorganism which may spoil the food and which are beneficial for the food. Okay, So the spoilish microorganism which will be spoiling the food, beneficial microorganisms are the type of microorganism which are used to produce the fermented food. Okay, so and you need to study about all these things in the food microbiology. You need to know about the growth curve of the microbes which you are using to in the fermented food. And foodborne disease, again very important. The type of bacteria which are affected, because generally it is bacteria or fungus only. So you need to know like which type of bacteria affect which type of food. Okay, so foodborne disease, what are the type of foodborne disease? Okay, that's important you to study. And again this part carries 3 to 4 marks, 3 to 4 questions, I'm sorry. And the last is about the food engineering. This is basically a kind of physical thing you need to know. So here how a mass transfer happens, how the heat transfer happens, okay, how molecular diffusion to take place. So say for example, how the gases moves from liquid, from air to the liquid form, solid state transfer, thermal transfer, all this thing you need to study over here in the food engineering, and it comes maybe under fermentation technology as well. So this part is generally covered into fermentation technology. So again, that you need to study about all this. And this molecular diffusion and fixed law are very important. The steady state mass transfer. So you can pay a little bit of attention to this. These are the important topics. So this is it. And the books for the food technology are Principles of Fermentation by Whittaker. Very good book. And again Food Microbiology by Fraser. Again very good book to be considered. Okay. These two books cover most of the food technology. Okay. If something is not there, you can refer to any other. But most of the part will be covered in this book. Okay. So this is it. So these are some tips and strategies how you can prepare the life science portions. Okay. That includes chemistry, botany, zoology, food technology, biochemistry and stuff. Okay. Thank you so much.
Our expert, Dr. Priya, and I hope you had a wonderful evening and you all enjoyed the sessions. I'm really thankful to each one of you who attended till the end, and we will like to come up with more such webinars in future. All your feedback and appreciation is highly appreciated. And just wanted to remind you, we are starting a fresh batch for CSIR NET and GATE exam from 1st of August 2019. In case you are interested, you can always join. Remember, the toppers are not built by chance. They are built by hard work. Also remember that if you sweat in peace, then you will not bleed in war. GATE exam is the war, but you need to sweat in peace right now. You have 8 months time to prepare for it, and Biotechnica is committed to help you. Okay, so help us help you by joining this batch, which is starting on 1st of August. Okay, and I hope you liked all the sessions. The recording of this session will be emailed to you today, and we'll try to send you today, if not today, otherwise tomorrow. Okay, I'll be sending you a feedback email. Please reply to that and share your feedback and suggestions. And yes, like I told you in chat, you can ask all your queries after this also in Ask Biotechnica. So if you just go and Google Ask Biotechnica, or you can go to this link, you can ask your questions, and all our people will be right there to answer queries. Yes, Preetha, if you've already joined, that's really great. We will send you the links. It will be available in your study portal as well. Also, just wanted to let you know, in case you have applied for the Schrodinger workshop, which is scheduled for 22nd of this month, the entrance exam has already started. You must have got an email to attempt that. It's a free workshop again with hard copy certificate. It's a tie-up between Biotechnica and Schrodinger. So if in case you want to attend that computer-aided drug discovery workshop, you're most welcome to do that. So that's it from now from Biotechnica headquarters. It was really a pleasure talking to all of you. Have a great evening ahead. See you soon in our next workshop. Thank you. Good night.